Hi, everyone. My name is Ishika Luthra, and I am a PhD candidate in the DeBoer Lab at the University of British Columbia. And today I'm going to be telling you about why we think regulatory activity is actually the default DNA state in eukaryotes. So I'm going to start by talking about what actually does the term functional mean in terms of biology. So we can think of it like if we have a piece of DNA that's changing the biochemical activity that then goes to downstream affect the fitness phenotype of an organism, we can annotate this genomic region as functional. Now, usually these regions have very strong conservation across species, which means that selection is acting to ensure that these regions are not mutated. Now, this doesn't actually work in the reverse order. So this doesn't mean that if you have biochemical activity or you're seeing biochemical activity, that it necessarily will change the fitness phenotype. So therefore, I would like to say that using biochemical activity alone is probably not a good marker of function. But biochemical activity does create a lot of molecular phenotypes. So we'll see things like co-occurring chromatin marks, we'll see variants that change gene expression, and then we'll see cell type specificity occurring because of these different genomic regions. But not all of these effects actually change the fitness of the organism. And I think one really good example of this is long non-coding RNAs. And people have seen that some of these do function in a tissue-specific way, and we have clear annotations for functions for some of them, but not all of them. So we're not sure what number of them are actually doing something important. So back in 2012, the ENCODE consortium proposed that, well, 80% of our genome had biochemical function. But like I've been telling you guys, this definition of function is a really long debated topic, and people use these different benchmarks for deciding what proportion of our genome is functional or doing something. Now, this makes it really difficult to assign a specific percentage to what proportion is functional without knowing the baseline activity of the genome without this constant selective pressures being applied. Now, this is really challenging since we know our genome is constantly undergoing evolution, there's random mutations occurring through it, and natural selection always acting on it. So the goal of this project was to be able to create a genomic null hypothesis in the absence of selection to help us get a better understanding of the definition of function in the biological context. So here we propose to use random DNA as a way to calibrate our expectations of what our genomes are going to do if there's no selection acting on it at all. And we aren't the first people to propose this idea. In 2013, Sean Eady suggested building a negative control using this idea of the random genome project, suggesting that we could use exogenous or evolutionarily distinct DNA, or like in this case, completely random DNA to create this null hypothesis. And since then, people have started doing this using reporter assays in yeast, in human, in Drosophila. And what they do is by inserting these small chunks of random DNA into cells, we see this cis-regulatory activity that shows that we can see this gene regulation occurring just by chance. Now, I'd like to point out that these studies are usually limited by the length of the sequence that we can put into the cell, meaning that we lose a lot of context of what's going on. So in this project, we're going to examine the baseline hypothesis in both yeast and in humans. And in yeast, we're going to do this using a yeast artificial chromosome, or a YAC. And in humans, we're going to do this experimentally using a machine learning model called Informer. So we actually can't feasibly synthesize really long stretches of completely random DNA, usually no more than 200 kilobases long. So instead, we wanted to use a yeast artificial chromosome, or a YAC, to introduce about 1.5 megabases of exogenous human DNA into yeast cells. And while yes, this DNA isn't completely random, we can kind of assume since human and yeast are separated by about 1 billion years of evolution, this DNA is random to the yeast, and it's a good proxy for evolutionarily naive DNA, since the yeast have never had to apply selective pressures to it. So we can take a look at the results once this work was done by a postdoc in our group, Cassandra Jensen. And what we find through RNA sequencing is, surprisingly, the yeast cell is actually interpreting the human DNA in a very yeast-like way. And what I mean by that is if we can take a look at this figure, at the top panel, you see about 22 kilobases of the yeast genome. And on the bottom, I'm showing you 22 kilobases of the human chromosome 7, so that yeast artificial chromosome. And from the figure panel, we can see that we see a very similar density of transcripts occurring between the evolved and the naive yeast DNA. We also see that the transcripts have similar length. Now, there are key differences. We find that naive or random DNA doesn't reach those very high extreme expression levels that the evolved sequences do. And this kind of suggests to us that evolution is actually necessary to reach these high expression levels. But overall, the results between the random and the evolved produce very similar transcriptomic patterns. And this kind of can take us back to our original question. We see all this activity, but just by seeing this activity, we can't say that this human yak had function in the yeast. So this leads us to the second part of the talk, which is, well, what is the baseline activity of the human genome? So like I just showed you guys, in yeast, we can test this question experimentally by delivering these long segments of DNA. But in humans, this process is very expensive and laborious. So currently, we wanted to use the next best thing, a deep learning model called Informer, which we've talked about a bit. But just briefly, so Informer can take in these up to 200 kilobase long sequences, and it'll predict the activity across 5,000 different chromatin marks. And the sequences we wanted to test here were 5 different sequences sets. So we used genomic sequences from the test set of Informer to avoid any trained test leakage. And then we wanted to put through completely random stretches of DNA matched to the GC content of the human genome. And then we wanted to test the effects of mono, di, and trinucleotide shuffling, both globally across the full 200 kb sequence, as well as locally in small 100 to 200 base pair wide windows. And what we find is very surprising. We find that this random DNA in human cells is predicted to be active, but it really requires that the local dinucleotide content is preserved. So what I mean by that is if we take a look at the panel on the left, and what I'm showing you on all y-axes here is the Informer prediction for one H1 DNA's track. And as we move from top to bottom, so the blue is the completely random sequence, and you can see there's very minimal regulatory activity there. And we see similar patterns in the global section where there's very little activity. Now, if we move to the right side, at the very bottom is the Informer prediction for an evolved region. And then as we look at the 3 tracks above with the local shuffling, you can see is that we do, as we maintain local di and trinucleotide content, we start to see this activity occurring again. And because we know that the genome has this really strong dinucleotide bias, but these sequences has never undergone any selection in the human cells, we can refer to these as naive to the human naive DNA. And instead of looking at this at a track by track basis, so just by one track, we can also look at this in like a more higher level. So here what we've done is we've clustered the predictions of Informer for 5 different chromatin marks, all for the same H1 cell line. And we cluster the 128 base pair Informer bins. And what we see here is that in the green, we have the naive or that dinucleotide shuffled sequences. Blue is those completely random, and then purple is those evolved sequences. And what we see is that the random sequences often cluster off by themselves, whereas you see the naive and the genomic have this co-localization or overlapping occurring. So this suggests to us that the sequences are more similar to each other than random. Okay, so if you remember at the beginning of the talk, I said people also use cell type specificity as a marker for function. So we wanted to see if this naive DNA also had cell type specific activity. So again, we're going to use both the genomic and the local shuffled sequences, and we predicted the DNA's tracks for 3 major cell lineages. So from the ectoderm, the mesoderm, and the endoderm. And the reason for doing this, the motivation for picking these 3 lineages, is because we know these lineages give rise to vastly different cells of the body, so are very unique from each other. And what I'm showing here is a scatter plot of the Informer predictions. Each dot is a prediction for 1 bin. In the purple, you see those evolved, those genomic sequences, and in the green, you have the predictions for these dilocal or naive sequences. And what we can see along the diagonal is that the naive predictions aren't just mimicking or recapitulating the genomic prediction. So this highlights to us that Informer hasn't just memorized some dinucleotide pattern, but it's actually predicting unique activity in different bins. We can also look at this between the naive and between the evolved sequences. And if we look at the upper right-hand corner, we can see that the correlations are lower than those for the evolved sequences. So this kind of tells to us that naive DNA is producing this more cell type specific expression than the evolved DNA. Now, instead of looking at this with just 3 different tracks, we can look at this across about 800 cage tracks from Informer. And what we did here is we calculated the track-to-track correlation for these cage tracks across all these different cell types. And what we see in the purple is that evolved correlations, and again, the green is the naive. And we see that the evolved correlations are shifted a little bit to the left, which is telling us the naive DNA yet again has produced this more cell type specific expression than evolved DNA, which again really brings us back to the original question: is using cell type specificity a good marker for function? So again, we're going to recall another marker for selection that is known is co-occurring chromatin marks. So we can look at this in two ways. So we can look at this from the t-SNE plots on the left, where I'm showing you again those same 5 H1 tracks from Informer. The t-SNE on the left panel is the genomic, and on the right is the naive. And what we see here is that many of the chromatin marks actually co-cluster in similar regions. So you'll see activity or high activity often occurring in similar regions of the clustering plots. We can also look at this like a bit more quantitatively by calculating the correlations between these 5 different tracks. So from the bar plot on the right, you can see that when we calculate the track-to-track correlations between these 5 tracks that we correlated, in some cases, the naive are a bit more correlated than the evolved, but generally we find that the results to be quite similar. So both by chromatin marks and the gene expression, this evolved DNA actually seems to be less cell type specific than the naive DNA in terms of its regulation. And since we see this type of pattern in the naive DNA, so DNA that has had no selection, we can once again bring us back to this may not be a reliable marker of selection. But we do understand that this was a significant domain shift for Informer. Informer has never been trained on random DNA. It's only ever seen genomic DNA. So we wanted to validate these results using a model that's only ever seen random DNA. So this model was published in 2018 by Sahu et al. And what they did here is they used a reporter system, or MPRA, in a GP5D cell line to annotate each 170 base pair fully random sequence as either active or inactive. They trained a convolutional neural network that would predict the probability of expression for each of these sequences. So what we did is we used the same genomic and naive sequences that we fed through Informer. We binned them to be of appropriate length, and then we predicted using this model that has no bias towards genomic DNA. And what we see is really interesting. We see that genomic models and these random sequence models produce really similar results. So I can walk you guys through this plot. On the x-axis is one Informer prediction track for a cell line matched to their study's cell line. And we binned the Informer predictions on the x-axis. And on the y-axis, you see the predicted probability of expression from this random DNA model. The red line is actually annotating the cutoff for where we would label a sequence as active or inactive. So above 0.5, you're active. And you can see as you move into Informer prediction bins that have very high predictions, so greater than or equal to 6, mostly all of the sequences in both the naive and evolved sequence sets will be labeled as active. And we can also look at the sequences, is Informer learning predicting relevant motifs? So what we did is, as a validation for Informer, and that the naive DNA wasn't just some sort of memory of Informer, we used simple enrichment analysis to find iPSC-related transcription factors in both these genomic and naive sequences. So from this plot, on the x-axis, you can see the log E value for the genomic motifs, and on the y-axis for those naive motifs. And each point is a transcription factor from the JASPAR database. And what's really interesting is we see 3 major pioneer transcription factors for iPSCs. So we see KLF4, NANOG, and MYC. And we do see SOX2 and OCT4, but they are not significant in this case. But this highlights to us that Informer has learned this transcriptional grammar. This was really encouraging to us. So this leads me to my final slide. Maybe, just maybe, not everything our genomes do is actually functional. We can conclude here that simply by seeing this transcription occurring, seeing co-occurring chromatin marks or cell type specificity, it's not enough to imply that a genomic sequence is functional. This baseline activity actually might reflect the need for our genomes to be evolvable. And the future directions for this is, as these synthesis technologies improve, we want to be able to validate these results in human cells, which will allow us to confirm our predictions that we see here experimentally, but also would allow us to train models using only DNA, larger models, so that overfitting to the genome is never an issue. We have a preprint out that you could take a look at for more information. And finally, I would like to thank the organizing committee here, as well as my supervisor, Carl DeBoer, my co-authors, Cassandra, Amelia, Rafi, and Aspar, and I'll be happy to take any questions.
Well, thank you, Ishika, for this very interesting talk. Questions? I wonder if you applied any interpretability methods for the prediction of informal on the random sequences to shed more light on what's going on and why is it happening?
Yeah, we would love to. I think we've been thinking about this a lot. So Informer is pretty difficult to interpret because of its transformer-like architecture. I know people are working on things like this, but that would be the end goal, would be to interpret what's looking at. That's kind of what we tried to do with the motif analysis, but yeah, currently interpreting Informer is pretty tricky from my understanding. Yes.
Okay, so Luca Pinello, please come up and put up your slides so we can, have another question. But we can just stand on the side of the microphone. Is there another question?
Did you load? I send the slide by mail.
I can just maybe talk. I've got a question essentially about the functionality and how people have been looking at pervasive transcription when you have, for instance, RNA degradation mutants, and is this something that you could also imagine using random sequences to look at RNA stability?
Yeah. Yeah, that would be really interesting. So doing some sort of similar to the Sahu et al. Model, having the predictions for both a random sequence as well as a base mutated, a couple bases mutated, that'd be really interesting, and I think it would help for sure. Thanks.